Hi everyone, I'm Dr. J from Department of Physiology, University of Malaya. I'm going to talk about mucosal protection in the gastrointestinal tract in this hour period. These are the lecture outcomes for this lecture period. We hope after this lecture you are able to describe the functional anatomy of the stomach, describe the GI protective mechanisms, describe the repair process that help to maintain the mucosal functions, and describe the consequences of impaired gastrointestinal mucosal integrity. Look at the gastrointestinal tract. This is the esophagus, stomach, small intestine, and large intestine. This one is a proper GI tract, and then the lining is covered by your mucosa. In addition to this proper GI structure, we also have associated GI glands, such as liver and the pancreas. Liver can secrete bile, which contain bile acids. Our stomach also secretes acid, or actually irritate our mucosa. And this structure is open up to the outer environment, so it's very important for our GI structure or mucosa to be protected against noxious substance and microorganisms ingested together with the food. And if you look at the small intestine, we have a lymphoid tissue that we call it pair patches. These pair patches also involving immunological reactions, and then is very important in protection against infection and inflammation. That's why our GIT is considered as one of the immunological organs in our body. Different part of the gastrointestinal epithelium has a different function. The esophageal epithelium is mainly composed of squamous cells, and then it has a protection function. Gastric mucosa, it has a mucus secretory gland, so it's for protection, and then it also secretes acid for digestion process. And we have an intestinal mucosa in the small intestine, so most of the intestinal glands secrete mucus, and then is for protection and also involving digestion and absorption. Whereas the colonic mucosa can secrete mucus and involving absorption of water for a certain amount, and then we have a lot of host bacteria in the colon, and then it causes fermentation and so on. Here's the gastrointestinal mucosa. It has three parts: epithelium, laminar propria, and the muscularis mucosae. Beneath the mucosa is a submucosal layer, and after that is a muscle layer, and outer layer is covered by serosa. Different type of glands present in different part of the GIT. The esophageal glands secrete mucus. The gastric glands are the single tubular glands and open into the lumen. So the opening, we call it gastric pit, and the main secretion is hydrochloric acid. The small intestinal mucosa folded like a finger-like projection. It increases the surface area for absorption. And then colonic mucosa has a colonic glands that secrete mucus. Okay. And in addition to this structure here, we have a special type of nervous system present in inside the wall of GIT. So inside the submucosal layer, we have one nerve plexus, and then in between two muscle layers, we have another nerve plexus. These nerve plexus are called enteric nervous system. The detailed function of the enteric nervous system I will discuss in next lecture. The mucosal integrity is maintained by GI defenses and offenses. These are the GI structure that can expose to acid. The main structure is stomach, acid secrete, hydrochloric acid. Usually, esophagus is free of acid because of the lower esophageal sphincter at the exit of the esophagus, which is tonically active and always closed. But in case this LES is not competent, the acid from the stomach can regurgitate back to esophagus, can irritate the distal esophageal mucosa. The acid from the stomach also go to small intestine along with the gastric emptying. In addition, the small intestine also can expose to the bile acids from your bowel. The large intestine, we have short-chain fatty acids and lactic acid from bacteria metabolism. Under normal condition, we have a laminar defense system to protect against acid. We have three types of defenses here. Pre-epithelial defense, which is our mucus layer to neutralize the acid. And we have epithelial defense, means our epithelial cells itself act as the physical barrier. And we have a sub-epithelial defense or post-epithelial defense, which comprise of blood flow and also the immunological cells. The epithelial defense, the mucus pincap layer, is a biochemical barrier and considered as the first line of defense against luminal aggressor. Two types of mucus layer is here. The inner layer is very firm and adherent and cover the whole epithelial surface. And the outer layer is a loosely adherent layer. Even though it covers the whole GI tract, actually the thickness is different from structure to structure. The outer layer, loosely adherent layer, is very thick in colon, whereas the inner firmly adherent layer is comparatively thicker in the gastric mucosa. And this layer acts as the diffusion barrier against the noxious agent. It acts as a lubricant, or it entraps the microorganism and interacts with the immune system. As the mucus is important in mucosal protection, you need to know more about mucus. It's a fluid secreted from our goblet cells. It's enriched in using glycoproteins that form large net-like polymers. This feature is obtained from rapid stomach mucosa. As for chemical composition, it contains water is around 90 to 95%, mucins is around 5 to 10%, electrolyte constitute around 1%, and it contains others like triphosphatase, enzymes, plasma protein, secretory immunoglobulin A, bacteria, and their decomposition products. The triphosphatase are recently discovered, and are produced and secreted together with mucin. The real physiologic role of triphosphatase are still under investigation, but we think it is associated with mucus and may improve its resistance to noxious agents. The high concentration of triphosphatase are seen at the site of injury, thus it may implicate in cell migration and cell repair process. A second line of defense is made up by our epithelial cells itself. The epithelial cells set up the physical barrier. At the apical surface of the epithelial cells, there are tight junctions. As its name is very tight. So the harmful substance from the lumen cannot go into the interstitial space. Here, there are a lot of plasma membrane exchanger and buffer. In addition, These cells, let's say if the cells undergo injury, these cells can heal itself. The process we call cell restitution. And these epithelial cells are under constantly renewal. Like us, it undergo aging, eventually die. All the dead cells slough off into the lumen and then replaced by the new one. It's cell replication. Post-epithelial defense mechanism also important. The mucosal blood flow supply oxygen as well as bicarb. In addition to this one, we have a local synthesis of prostaglandin here. This prostaglandin is very important as it stimulates the bicarb secretion from your epithelial cells. And then we have a mucosal nerve, and as I told you, in the small intestine, we have lymphoid tissue here. All the immune cells can protect against the infection. Move on to the mucosal protection in the individual structure in the GIT. Let's start with esophagus. This is your esophagus. Esophagus is a tubular structure. At the entrance of the esophagus, we have one door. This door we call it upper esophageal sphincter. And at the exit to the stomach, we have another door that we call it lower esophageal sphincter. As you can see here, the upper part, the proximal part of the esophagus is actually exposed to saliva. So we have bicarb, epidermal growth factor, and prostaglandin E here. Okay, but the distal part of the esophagus seems to be more acidic because of the acid regurgitation from your stomach. So the mucosa, esophageal mucosal gland can secrete epidermal growth factor, prostaglandin E, and mucus. This is the esophageal mucus buffer layer. If you look at the pH immediately above the epithelial surface is around 2 to 3, whereas in the lumen is pH 2. Means this barrier can protect against approximately 1 pH unit change only. So seems to be weak, seems to have a weak protection. Why? It may be because of the lack of the well-defined mucus layer, and the bicarb has a limited permeability, and then the surface cell cannot secrete bicarb also. As the stomach acid can regurgitate back to esophagus, this lower esophageal sphincter is very, very important. The lower esophageal sphincter, together with the part of the crural diaphragm, set up anti-reflux barrier. The crural part of the diaphragm can receive nerve supply from Franklin nerve, which secrete acetylcholine from its nerve ending as a neurotransmitter. This blue nerve represents vagus. So this vagal activity regulates the LES condition. The vagus act on the intrinsic nervous system here. Remember, inside the wall of GIT, we have intrinsic nervous system. A lot of neurons present in the intrinsic nervous system. If the vagal activity activates the interneuron that secrete acetylcholine, it causes constriction. If the vagal activity activates the interneuron that secrete VIP, vasoactive intestinal peptide, and nitric oxide, it can cause relaxation. Usually, this anti-reflux barrier is tonically active. But relaxed briefly on swallowing or bulging. In the case of incompetent ileus constriction or transient ileus relaxation, the acid reflux occurs. And acid can regurgitate back to distal esophagus. H plus can go in through the dilated intercellular space and cause epithelial injury. This condition is called non-erosive reflux disease. Can cause inflammation, later on can lead to erosive esophagitis and end up with Barrett's esophagus. If the acid reflux can cause symptoms or complications, it can lead to gastroesophageal reflux disease, GERD, usually associated with two syndrome. Different esophageal syndrome means symptoms are confined to esophagus. Symptomatic syndrome with typical reflux syndrome, or sometimes syndrome associated with the esophageal injury. In the extraesophageal syndrome, established conditions like reflux cough, reflux laryngitis, reflux asthma, reflux genital erosion, and sometimes associated with proposed conditions like sinusitis, pulmonary fibrosis, pharyngitis, and recurrent otitis media. Let us move to the mucosal protection in the stomach and duodenum. This is the stomach. It can be divided into fender portion, corpus or body. Here is the antrum, and the one near to the duodenum is the pylorus. Okay, so this is the duodenum. It's the first segment of the small intestine. Okay, so the stomach content can go into the duodenum. Look at the acid gland. This is the single tubular glands. So a lot of lining cells are here. The chief cells are mainly secrete pepsinogen. Here we have an ECS cell for histamine secretion. These green cells can secrete parietal cells. These parietal cells are also called auxantic cells. It can secrete acid and intrinsic factor. Here are the mucous neck cells, and these are the surface mucus cells that secrete mucus, triphosphatide, and bicarb. So all open up into the lumen. This opening is, we call it gastric pit. Gastroduodenal defense mechanism is set up by gastroduodenal mucosal barrier. The surface mucus cell secrete mucus and bicarb. So this mucus form a protective gel-like layer over the entire surface. So the inner atrial layer is very, very thick, whereas the outer layer is a little bit thin. And in between, bicarb is trapped. So the bicarb secretion is active process and activated by your vagus stimulation, fender distension, and prostaglandin E series. So here, because of this mucus and bicarb, the pH immediately over the apical surface is around 7, whereas in the lumen is around 1.5. So we can say this gastroduodenal mucosal barrier is very strong against acid. As usual, this defense comprises of pre-epithelial defense. Just now I mentioned the mucus layer, and then we have an epithelial defense set up by cell renewal. The stomach cell also always constantly under constant renewal, and the prostaglandin stimulate our bicarb secretion. And then in the post-epithelial layer, we have microcirculation and the nerve supply that stimulate the continuous oxygen supply and the nutrients and so on. I'm sure you noticed about the role of prostaglandin in GI defenses. Let me summarize the protective effects of prostaglandin. Prostaglandin stimulates the mucosal cells to secrete bicarb. It also stimulates the mucus secretion. It stimulates the mucosal blood flow. It stimulates the restitution and proliferation process. And it reduces the hydrochloric acid secretion from parietal cells. Alright, so we have protective healing factors such as bicarb, prostaglandin, mucus, and the mucosal blood flow, whereas we have ulcerogenic factors. The bad guys are acid, Helicobacter pylori, pepsin, nonsteroidal anti-inflammatory drugs, NSAIDs, and the smoking, alcohol, stress, several drugs. Okay, so usually under physiological condition, these protective factors, the good guys and the bad guys, are imbalanced. But if an imbalance between hostile and protective factors, it can lead to breakdown of gastric mucosal barrier. Impaired mucosal integrity can lead to several consequences. The mild impaired mucosal integrity can result in erosion only, whereas the serious one can cause ulcer. If the ulcer involves the serosa, it can lead to perforation. There are many causes of peptic ulcer. The main contributors are NSAID and H. pylori infection. The NSAID mainly cause gastric ulcer, whereas the H. pylori are mainly occur in the duodenum. And in addition, the alcohol, smoking, drugs, medication, all can cause erosion and peptic ulcer. So if we say peptic ulcer, it can be either in gastric or in duodenum. As a medical student, you must know the role of NSAID. The NSAID are prostaglandin inhibitors. So basically, we have two types of prostaglandin, physiologic prostaglandin and pathologic prostaglandin. In the last case, we use traditional NSAID that can inhibit both physiologic and pathologic prostaglandin. But nowadays, we have a COX-2 inhibitor that inhibits only pathologic prostaglandins. These are several mechanisms that NSAID can cause gastric injury. As I told you here, the NSAID, if you inhibit the prostaglandin, means the impairment of mucosal defense and repair. So that's why the mucosal barrier can be broken down. And NSAID also inhibit the cell proliferation and also inhibit the angiogenesis, so impairment of GI repair, and then can cause GI bleeding by inhibition of thromboxane, and it acts as an irritant, and it damages the blood vessels, and it has the various effects like inhibition of other enzymes like phospholipase, reactive oxygen species, interact with the inducible nitric oxide synthase and direct effect on genes. With the discovery of H. pylori, the knowledge on gastric pathology has been extended. In 1982, Australian Dr. Barry Marshall and Dr. Robin Warren discovered H. pylori as a causative agent for pathogenesis of chronic gastritis and gastric ulcer. It is a gram-negative, helically shaped microaerobilic bacterium. It has an outer layer, and then the outer layer has a protein and lipopolysaccharides to adhere the host cells. And it also contains exotoxins and secretory enzymes that can cause injury. Okay? And it has a flagella to move around. And like other bacterium, the H. pylori can overcome the stomach acidity. Because of its characteristic, it can produce large amount of urease. Once the H. pylori enters to the stomach, it adheres to the gastric epithelial cells and produce a lot of urease. In a subsequent urease-driven enzymatic reaction, the urea is converted to carbon dioxide and ammonia. And because of that, hydrochloric acid is neutralized and then pH is high. Okay? So the gel-like mucin transition from gel to soluble form, elasticity is reduced. So more H. pylori are swimming freely through the mucus and adhere to the epithelial cells by the action of outer proteins and lipopolysaccharides. And then it starts to secrete exotoxins and secretory enzymes, mucinase, protease, lipase, all can produce gastric mucosal injury. These are the consequences of H. pylori infection. Once the H. pylori infection occurs, can lead to superficial gastritis, atrophic gastritis, and undergo intestinal metaplasia or spasmodic polypeptide expression metaplasia, lead to dysplasia and gastric adenocarcinoma. Of course, it's associated with the risk factors such as diet, bacterial virulence, smoking, and host genotype, such as stroma remodeling, inflammation, apoptosis, and proliferation. Look at the mucosal protection in the small and large intestine. This is the small intestinal mucosa. So the mucosa is folded, and then we call this structure as villi. Here is the colonic mucosa. So both small intestine and large intestine, it has glands, and then stem cells are present in the bottom of the glands is for cell renewal. So the mucosa has goblet cells to secrete mucus, but in the large intestine, the goblet cell number are very high. That's why it's responsible for the thicker outer mucus layer. Remember, large intestine has a very high mucus layer, right? Okay. In addition, the small intestine, we have a lymphoid tissue, and then we have microphore cells. We call it M cells. As usual, we have three defense layer here. Pre-epithelial mucus layer. The outer layer is the layer where the commensal bacteria can be trapped, and the inner mucus layer acts as the biochemical barrier. The intestinal cell itself is acts as a physical barrier. And then we have immune cells in the laminar propria. This is like an immunological barrier. Here, like macrophage, dendrite cells. Okay. So, in addition, these cells, some of the cells in the intestine can secrete antimicrobial peptides, also for protection. In disease pathogenesis, this intestinal barrier can be broken down. If the barrier broken down, in some cases, it restores homeostasis by epithelial regeneration, decreased immune activation, and restoration of the tight junction barrier. But in some cases, this one cannot be restored. So probably the tight junction are loose, cell undergo apoptosis, and then so once the tight junction are loose, all the harmful substance from the lumen can go in and then activate the immune system and leading to the disease process. A breakdown of intestinal barrier function usually associated with the inflammatory bowel disease. We have two types, Crohn's disease and ulcerative colitis. So here, the microbiota or the aggressors can activate the immune response. In the submucosal layer, here the cytokines are activated and leading to inflammatory bowel disease. The detailed explanation of this IBD will be discussed in other lectures. With the advancement of medical technology, now we can visualize the inner lining of gastrointestinal tract. The procedure is called endoscopy. As the name implies, it means to look inside. In this procedure, long flexible fiber optic tube with the tiny camera at the tip is used. This endoscope is inserted to the gastrointestinal tract, and then the camera captures the image of the GIT. Two types of endoscopy here. Upper GI endoscopy we call esophagogastroduodenoscopy, EGD, can visualize the upper part of the gastrointestinal tract down to the duodenum. In lower GI endoscopy, the sigmoidoscopy can visualize the sigmoid colon, whereas the colonoscopy can visualize the whole colon. I hope this lecture will help you have a better understanding on mucosal protection in the gastrointestinal tract. If you have any questions, please note down. We can discuss during live discussion session that will be conducted using Microsoft Teams. Thank you.